Hello dear students, this is Tanveer Malik with a new video lecture. In this video lecture, we shall be discussing about the digestion of proteins. In the previous lecture, we had discussed about the digestion of carbohydrates, and in this particular lecture, we shall be discussing about the digestion of proteins. First of all, we should be able to know what actually proteins are. Proteins are actually complex compounds of amino acids. So proteins are actually the complex compounds or amino acids, or we can say simply that proteins are made up of amino acids. And when we talk about the digestion of proteins, when we carry out the process of digestion in our body, it's not only that at a moment or in a single step that these proteins get converted into amino acids, because proteins are made of amino acids. When we will carry out the process of digestion of proteins, that means at the last stage, we must get amino acids. But these amino acids are not obtained at one go. First, these proteins will get converted into large peptides, and then these large peptides will get converted into dipeptides, and finally dipeptides will get digested into amino acids. So when the process of digestion of protein takes place, first we shall get large peptides. Large peptides will further get digested into dipeptides, and dipeptides will at last get converted into amino acids. But we must know what are actually peptides, what are dipeptides. Large peptides are actually smaller proteins. These are small sized proteins, and they are made up of about 3 to 50 amino acids. So large peptides are small sized proteins. They are made up of 3 to 50 amino acids. And when we talk about the dipeptides, these are made of 2 amino acids. These are made of 2 amino acids. And these 2 amino acids can be similar or they can be different. So let us see how exactly the digestion of protein start in our digestive system, in our body. First of all, when we take the protein-rich diet in our mouth, in our oral cavity, there is no digestion of protein in the oral cavity. Why? Because there is no protein digesting enzyme in our oral cavity. In the previous lecture, we had seen the digestion of the carbohydrates started from the oral cavity. But the digestion of protein does not take place in the oral cavity because there is no protein digesting enzyme. So when we take up the proteins, they are directly sent into the stomach through esophagus, and it's the stomach where the process of the digestion of protein starts. Actually, the stomach, we all know that the lining of the stomach contains some essential glands which are called gastric glands. And what does these gastric glands secrete? They secrete gastric juice. They secrete gastric juice. And this gastric juice contains HCl, hydrochloric acid. It contains some proenzymes. It contains HCl and it contains some proenzymes. And what is the function of HCl? If there are certain microbes present in the food, this acidic medium, this hydrochloric acid, is going to kill those microbes. And what are the proenzymes present in the gastric juice of the stomach? There are 2 types of proenzymes. First one is pepsinogen, and second one is prorenin. So we have got 2 types of proenzymes in the gastric juice of the stomach, pepsinogen and prorenin. But before we see the function of the pepsinogen and prorenin, we got to understand what are actually the proenzymes. Proenzymes are actually the inactive enzymes. These are the inactive enzymes. Why are these inactive enzymes? Because if they are active at the first stage, they are going to digest the cellular contents of our body. They are going to destroy the cellular contents of our body. So these are only activated at the site of action. When the action is going to take place in the stomach, that means the digestion of the proteins, when the digestion of the proteins is going to take place in the stomach, at that particular moment, these enzymes will get activated. And how these get activated, we shall see that. For example, we have got the pepsinogen enzyme. This pepsinogen enzyme is actually a proenzyme. It is an inactive enzyme. This inactive enzyme is activated by the HCl, hydrochloric acid. And this hydrochloric acid converts this pepsinogen into pepsin. Now the pepsin is the active enzyme, and it is this pepsin which can act upon the proteins. Similarly, prorenin is a proenzyme. HCl will again act on the prorenin and it will convert it into renin. So pepsinogen and prorenin are the proenzymes. These are inactive enzymes. They are not going to digest the proteins unless and until HCl acts on both these proenzymes to convert them into the pepsin and the renin. And it is this pepsin and renin which is actually going to carry out the digestion of the proteins. Now we shall see how does this pepsin and renin digest the proteins. Because I have already told you, in oral cavity no digestion of protein takes place. It's the stomach where the actual start of the digestion of the proteins take place. So we shall see. For example, we have taken a protein-rich diet and these proteins have reached to the stomach through esophagus. Now these proteins will be acted upon by the pepsin. And this pepsin enzyme will convert this protein into large peptides. I have already told you that proteins are not going to be converted into the amino acids in one go, in one moment, in one reaction. They first will be converted into peptides, then dipeptides, and finally into amino acids. So what are we getting? Pepsin, the enzyme in the stomach, is acting upon the proteins and is converting them into the large peptides. Similarly, what does this renin do? Renin acts upon the Malik protein. And that Malik protein is called casein. Renin acts upon this casein and it converts it into paracasein. Paracasein. But this renin enzyme is only present in the stomach of the kids, infants. But it's absent in the adults, because in the adults the Malik protein is digested by the pepsin. So this casein protein is only found in the stomach of the infants, and in the stomach of the infant this casein protein is acted upon by the renin and converts it into paracasein. And after that this paracasein combines with the calcium ions in the stomach and gets converted into calcium paracasinate. Calcium paracasinate. And after that, what happens to this calcium paracasinate? I will write here. Calcium paracasinate will be again acted upon, not by the renin, but by the pepsin. And it converts this calcium paracasinate into the large peptides. So what actually happens in the stomach? We have got the gastric glands of the stomach that produce gastric juice, and gastric juice contains HCl and proenzymes. Two proenzymes, pepsinogen and prorenin. What does this HCl do? It has got the antimicrobial activity. It will kill the microbes present in the food. And besides that, the proenzymes, the two types of proenzymes, pepsinogen and prorenin, these are proenzymes, they are not going to act upon the proteins because they are inactive. They will be activated by the hydrochloric acid. HCl will act upon the pepsinogen, convert into the pepsin. HCl will again act upon the protein, convert into the renin. So after that, this pepsin and renin actually start the process of digestion of the proteins. Pepsin acts upon the protein, converted into large peptides. What are peptides? Peptides are the smaller sized proteins. They are made up of 3 to 50 amino acids. After that, what does this renin do? Renin acts upon the Malik protein, casein, which is only present in the stomach of the infants, and converts it into paracasein. Paracasein combines with the calcium to form calcium paracaseinate in the presence of renin enzyme. After that, calcium paracaseinate will not now be acted upon by the renin, but it will be acted upon by the pepsin enzyme and will convert it into the large peptides. So in the stomach, we finally have got large peptides. Whether pepsin acts on the proteins, large peptides. Whether renin acts on the proteins, large peptides. So we have not yet got the amino acids. After that, what happens? This food, these proteins which have been converted into large peptides, they will get into the small intestines. Now the second step in the digestion of the proteins is that this process will now start in the small intestines. Small intestines. Small intestines, in the previous video lecture, I told you that intestines contain two types of digestive juices. First one is pancreatic juice and second one is intestinal juice. Intestinal juice is produced by the small intestines themselves, while as pancreatic juice is received by the small intestines from an important organ, that is pancreas, through hepatopancreatic duct. So this pancreatic juice when reaches into the small intestines, how does it help in the digestion of the proteins? Actually, this pancreatic juice contains so many enzymes which actually carry out the digestion of the proteins. What are those enzymes? We have got trypsinogen, we have got chymotrypsinogen, and we have got procarboxypeptidase. So there are three types of protein digesting enzymes in the pancreatic juice. You will remember that all those enzymes which carry out the digestion of the proteins are called proteases, or they can also be called as peptidases. So all the protein digesting enzymes are called proteases or peptidases. In the last video lecture, I told you all those carbohydrate digesting enzymes are called carbohydrases. Similarly, protein digesting enzymes are called proteases or peptidases. So let us come back to the pancreatic juice. It's containing three types of enzymes: trypsinogen, chymotrypsinogen, procarboxypeptidase. But again, you will remember that all these three enzymes are again proenzymes. These are again proenzymes. That means these are inactive enzymes and they are not going to digest the proteins unless and until they get activated. Now we will see how exactly these proenzymes are activated. First of all, we shall take the example of the trypsinogen. This intestinal juice contains an enzyme called enterokinase. The intestinal juice contains an enzyme called enterokinase, and it is this enterokinase, because pancreatic juice, intestinal juice, both are present in the intestines, and intestinal juice contains enterokinase. This enterokinase enzyme actually acts on the trypsinogen, which is a proenzyme, and converts it into trypsin. So trypsinogen proenzyme is converted into the active form, that is trypsin. Which enzyme carries out this process, carries out this reaction? It is enterokinase present in the intestinal juice. Now, once the trypsin is formed, now this trypsin will activate these two proenzymes, that is chymotrypsinogen and procarboxypeptidase. This trypsin will act on the chymotrypsinogen and will convert it into chymotrypsin. Similarly, this trypsin will again act on the procarboxypeptidase and will convert it into the carboxypeptidase. So we had three proenzymes of the pancreatic juice: trypsinogen, chymotrypsinogen, procarboxypeptidase. Now we have got the active form of these enzymes. Now these active forms of these enzymes will carry out the digestion of the proteins. But what actually we have received from the stomach? We have received large peptides from the stomach. But not all proteins have been converted into large peptides in the stomach. Only a certain proportion of the proteins have been converted into large peptides in the stomach. But there is a lot more proportion which is still not converted into large peptides. And we shall see how exactly these three enzymes carry out their reaction. So we have seen that not all the proportion of the proteins are converted into the stomach. There is a lot more proportion still left which is not converted into large peptides. So we have got still proteins which have not been converted into large peptides. So those proteins, well, they will enter into the small intestine. These proteins will be acted upon by the trypsin. Sorry. And this trypsin will convert these proteins again into large peptides. Still not all the proteins are converted into large peptides. Still some proteins are left which are not converted into large peptides. Those proteins will be acted upon by the chymotrypsin. And chymotrypsin will convert these proteins into into large peptides. So at this particular moment, all the proteins now have been converted into the large peptides. A certain proportion of the proteins was converted into large peptides into the stomach, and rest of the proportion is converted into the small intestines into large peptides by the action of trypsin and chymotrypsin. Now what does this carboxypeptidase actually do? It will act upon these large peptides. So we have got large peptides from the stomach and large peptides formed in the small intestines. These large peptides will be acted upon by the carboxypeptidase, and they will convert these large peptides. When carboxypeptidase acts on these large peptides, we get dipeptides and some amino acids. But you got to remember that not all the large peptides are converted by carboxypeptidase into dipeptides plus amino acids. There are still a larger proportion of the large peptides which are not yet converted into the amino acids. So this is all the action of the pancreatic juice and its enzymes on the proteins. So after the action of the pancreatic juice, we have got dipeptides plus amino acids, but there is still a larger proportion of the large peptides which are not yet converted into dipeptides and amino acids. And which enzyme actually converts those large peptides into amino acids? Still the action of the intestinal juice is pending with us. We shall see what does this intestinal juice actually do. Intestinal juice contains two types of enzymes. First one is aminopeptidase and second one is dipeptidase. But you have to remember that the aminopeptidase and dipeptidase, these are not the proenzymes. Remember, these are not the proenzymes, these are the active enzymes. So we don't need to activate them. They are already in the activated form. So whatever the large peptides are still pending here, whatever the large peptides have not been converted into dipeptides and amino acids, those will be acted upon by the aminopeptidase. So I will write here large peptides. These large peptides will be acted upon by the aminopeptidase, and aminopeptidase will convert these large peptides into dipeptides plus amino acids. But so at this particular moment we have seen that all the large peptides have been converted into dipeptides and amino acids. Carboxypeptidase has converted certain proportion of large peptides into the dipeptides and amino acids. Now aminopeptidase is converting another proportion of large peptides into dipeptides plus amino acids. Now what does this dipeptidase enzyme do? Still we have got dipeptides which are formed here and dipeptides which are formed here. These dipeptides will be acted upon by the dipeptidase enzyme, and it will convert them into the amino acids. So finally, this process of the digestion of proteins has got completed. There was no digestion in the oral cavity because of the lack of the protein digesting enzyme. Digestion of proteins started from the stomach and it ended in the small intestines with all the amino acids in the small intestines. These amino acids will get absorbed in the blood and they will move into the cells where they will be used for the growth and development of the cells, growth and development of the tissues, organs and the whole body. So if you think that you have understood this concept, don't forget to subscribe my channel 360 biology tutorials. Don't forget to press the bell icon to get the latest updates. Till then, thank you very much.